seem a little dark. I have bottom.
We have just sighted the bottom.
Sirius 25.
You got it.
Good video.
Oh, I'm getting pulled back. Can you keep
Looks
coming
like
down,
we have
please?
soft unconsolidated sediment again.
Coming down.
Can see our thrusters
I just got
causing a minor
tugged all
dust
over the
storm.
place.
It's generally true that we
That's
try to
good.
find a pretty flat landing site for D2. This avoids complexities related to ups and downs as we're situating ourselves to get ready for the dive. So we're
15.
approximately
Yeah,
at the base
same
of this
issue
slope
you did yesterday.
on a little bench.
I was going full opposite, and I was just getting
when we get set up,
yanked backwards.
we do that. As soon as we get set up, we'll turn and face the slope and begin our transit upslope.
Okay, let me come down. Sonar froze. Can you?
Sure.
Yeah, I think we've lost.
Looks like we've got some
ROV watch lead.
modeling. Go ahead.
Survey 1, ROV watch lead. Yeah, I don't think we're controlling the vehicle. You can watch when I put verticals down. Get a nice pitch and roll, or a nice roll when I do that.
In the distance on the right, we have our swimming holothurian again.
Still trying to set up here. We always have to check systems once we get to the seafloor. This is particularly true for thruster systems on the D2 when we're near a slope like this. This is a real dance when we're going upslope. We have to be very careful not to impact the biology and the geology we're looking at, but also not to inadvertently contact the slope, which can lead to problems with
red on and off
with the equipment. So got to check systems before we start
DC
moving
bus.
towards the slope,
right.
which is we're close to the base of the slope here.
Roger that. DC bus return. Briefly red.
All right, here we go. Motor coming on.
Still green.
It takes a few seconds to get any update.
It's red. Back to green.
Okay, will do. All right, Chris, in that case, we're already 90 out, so do you want to keep coming port?
Sure.
We have an issue with a vertical thruster on the D2, but we've been informed that that should not impact our maneuverability for this dive, so we're going to proceed as soon as we're set up, and that should happen here shortly.
Let's see, I need to turn. Hold on. Let me turn.
Roger that.
Co-pilot, I'd like to shut down high pack for just a second and restart it.
Is our tracking bad?
Just to refresh the waypoints.
Okay.
Should I hold off on that?
Yeah, can you hold off on that until we're done, moving here?
Sure.
Reset.
Reset.
Turned. Okay.
Water visibility looks relatively good today. We had excellent visibility on dive 1. Visibility was still good on dive 2, but we had a fair bit of snow in the water column. It looks like we've got some today. For those of you who had joined us yesterday, we had dive 2 prematurely shortened by sargassum weed clogging the main engine intakes on the Okeanos
Are you
Explorer.
backing down?
There's an extraordinary amount
No,
of sargassum at the surface in the Gulf
Come your direction.
this month for reasons unknown to me. If other people know why, I would like to know about it. We've seen a lot more sargassum at the surface here. We're diving a little southwest of where we were 2 years ago.
Much harder.
Maybe that's a factor, but there seems to be an extraordinary amount, and it gave us trouble yesterday. We haven't had problems yet this morning, but the engineers are going to be looking a lot more closely at intakes today. We almost had a shutdown of one of the mains yesterday, and we had to come off the seafloor prematurely. Not something we want to do again, so we're being very careful with it. As you know, when we're diving, the ship is in what we call DP mode, dynamic positioning mode, and is moving very slowly, if at all, through the water column. So the potential for encountering these windrows of sargassum and having them pulled into filters goes up. We hope that won't happen again. This is the Okeanos Explorer.
Yep. I'm not sure what that was in the Sirius.
Come left now.
ROV nav, are we headed for the base of this slope now?
We're still getting set up, but we will be heading upslope shortly after.
Roger that, thank you.
Lining up, it should be coming out right below you. Let me get down to the bottom.
Got you on bubble cam. PTZ.
Yep, and back down. All right, pushing out now.
Should be able to see you in the main HD here soon. Once I see you in main HD, if you're okay with it, we'll let Nav restart high pack.
As we're approaching.
Oh, there's a squid at the bottom. I'm sorry. Didn't mean to cut you off. Go ahead.
You alright with that,
Sorry.
pilot?
Yeah.
Go ahead, Nav.
Go ahead, Brian. It's fine.
I was just saying I want to be on the lookout for hard targets in the sonar, the D2 sonar. I see a couple, I think, coming through. If we can, as we go up, try to. I have a feeling that a lot of the slope is going to be sedimented, but with isolated patches of exposed hard bottom. So if we can just be on the lookout for those, and if possible, to do small zigzags to hit them, that'd be great.
Okay.
Yeah, we're looking at the sonar now, Brian, and we'll, as I said, we're in no rush to get up this slope. So we'll pretty much be guided by what you'd like to see and how long you want to see it.
I was just thinking that. Which one is that? Let's see. Not that one.
Water depth right now is
What, the one in your eyes?
1,153
Yeah.
meters.
Would be,
We are not quite yet
lower
at the base of
LED.
slope, but we will be
Lower
shortly. I'm
LED.
seeing it ahead of us on
Thank you.
on our nav display. Still probably 100 meters or so from the start of that slope. As I said earlier,
Pilots, are
it's
we
generally
underway
important
at this
for us
point?
to be
We are not.
deploying
Okay.
the ROV in Sirius at a flatter spot to get organized before we head up a slope like this.
And there's a swimming holothurian in the Sirius camera bouncing around.
I think it's probably the Sirius camera that's bouncing
Yeah.
around, not the holothurian.
Yes.
But yeah.
He's passing the screen quickly.
The Sirius camera sled, Stephanie points out, does feel the ship motion, and those of you looking at the Sirius view can see the tether that links D2, which is close to the seafloor, and Sirius. It's about a 30-meter tether, and we like to keep some slack in that tether for obvious reasons. We want to be able to move D2 around without feeling the tug of Sirius above it. Sirius is hanging on a much heavier duty tether from the ship, but it always feels a bit of the ship motion, what we call heave, and there is a bit of heave out there today. We've got 4 to 5 foot seas out there right now, pretty strong wind from the south, so we will see a bit of a vertical motion in Sirius as we go through the dive.
All right, Nav. Which way am I heading to waypoint 1?
So we're over waypoint 1 right now, and we're going to be heading upslope toward waypoint 2.
Okay.
So let's see, it looks like it's just about due north.
Yeah, that'd be fine. While we've got a moment here, it would be useful for everyone
Go ahead, Bridge.
to announce themselves, everybody who's on the event log, and let us know what your interest is in the dive and how
Okay,
you'll be
thank you,
helping
Bridge.
us through telepresence as we
Oh,
go
let's
through
know.
go through the dive. Brian Kinlan from NOAA, who nominated the dive, has already done that. Can we have some more people chiming in?
All right, we're back. Go ahead, Nav.
Don't be bashful.
Bridge just notified me that they moved their heading 5 degrees port.
Okay.
How about you, Bob? Carney, why don't you let us know what your interest
So
is?
waypoint 2 is at a bearing of 345,
No audio.
and it's just under
no audio. Well,
500
that's
meters away.
fine. Bob Carney's from
Copy that.
Louisiana State
And
University.
that is
He is a
our
well-known
goal for the
authority
day,
on
is
the
to
Gulf
hit
of
waypoint
Mexico and the
Okay.
biology
Go
of
beyond
same.
that if we have time.
Mike, are you
Okay.
with us? Mike
So
Vecchio?
we're climbing
So we're just going to
this
head right
gradient.
upslope. Yep.
Okay. So, Bobby, that's to both of our port.
Mike may not have. Who's out there that's
If you
got
just
audio
wanna
capability? We've got people at Stennis.
Lateral around and elsewhere.
The ECC at Stennis. Do you want to chime in and let us know who's there, please?
Is
Video
anyone
starts
else on the
going here.
conference call besides Brian?
Mike just chimed in on our event log that
Video?
he's got interests in the swimming biology that we're going to see today, especially squids and octopods. We have not yet seen. I guess we just saw a squid, but we have not seen octopi yet. There's a nice. Is that a tripod fish in the view now?
No, it's some kind of rat tail.
A rat tail. Okay.
He's quite beautiful. Look at his pectoral and dorsal fins are real long.
Getting closer to the base of the steeper part of the slope now.
Yeah, that's a wide video. I'm too far out.
And Brian, chime in if anything at all that you see that you want
Pushing
closer
me
looks
away
at.
from the fish.
Stephanie and I will do our
As
best
I bounce up
to
and down
accommodate
with
you and
only one vertical.
everyone
So
else on the dive
northeast
that's
to
interested in the
southwest.
biology we're going to encounter here.
Sort of.
Go ahead, video.
Small shrimp there
Yeah,
too.
some shrimps.
Yep.
There's another one swimming.
Chris, take
I may be the only one here. Ryan Kimlin from Hancock.
Go ahead, Ridge.
Coming up.
It's Steve
Oh.
Giddings. I
Oh.
just joined down in Galveston. I'm Tommy Heaton and Bill Keeney.
keep
Could you
going,
repeat that? I'm sorry.
Yeah, this is Steve Giddings. I'm down in Galveston.
Hi, how are you?
And,
Welcome to the line.
great, great. Thanks.
Is that something in his mouth?
No, it's just on the other side.
Looks like that rat tail is interested in a
Great,
shrimp
thank you.
meal.
He almost caught him.
All right, let's come wide. I'm bouncing quite a lot here.
Yeah.
Workshop, Nav.
Come down.
This is the first
Yeah,
time
you can-
But
this species been
That's
sighted.
I'm also
From
not
this
sure
Certainly
what I
ROV?
can
on
do.
this on this
Just
set of dives.
letting you know,
yes,
I got an
think so.
update from the bridge about the weather.
Roland is saying
Looks like
from
we're,
ever on the
holding steady at about 2
on
to
the
4
ship.
feet at the moment, but we'll keep an eye out for it for the rest of the day.
giving you your camera back.
Bill Keeney and Steve Giddis are joining us from the Galveston Exploration Command Center. The Sanctuaries program, which is part of NOAA, is also very interested in the kinds of dives we're doing here. We'll be doing some dives down at the base of Keithley Canyon and Bryant Canyon later on in this expedition. Those are two of the big sediment
Okay.
drainage pathways through this very complicated topographic zone of enclosed basins and broken ridges that form the north central Gulf of Mexico. And again, we'll be looking at biological habitats in a variety of water
Pilots,
depths
Nav.
on those dives.
Go ahead,
So
Nav.
we've got a lot of
Just let me
diversity
know
in the
when you're
dives
ready
that
for
are
a
coming
ship
up.
move.
Yeah, copy that.
What's our heading?
The next target's at a bearing of 345.
345. Okay.
We've already passed waypoint 1, correct? And we're pointed to head up slope. Here's another
I think
rat
we
tail out
landed
of
just
us.
about on waypoint 1. We're heading up slope.
Yeah.
Yeah, the ship's more or less on top of waypoint 1. The
just went off screen of
D2 and
10
Sirius
kilometers.
are a little bit slopeward of the ship at the moment. Another rat tail, I guess.
if we can image this
Yes.
guy.
Yeah, go ahead.
On the prowl for shrimp dinner, shrimp breakfast. Shrimp seem to be accommodating them by being close by. Of course, it's always good to remember, those of you who are sitting back watching us, that the light environment here is highly unusual for these organisms. They're not used to dealing with nice bright LED lights that we provide them as we go by. They're usually dealing with a pitch black environment and dealing with all of the needs of operating in a pitch black environment. We like visible lighting to help us observe these organisms, but that's not the typical mode in which they operate.
Let's move on, Roland. Go to your target over here.
What's that thing to the left? Is that just a stick? Looks like a crinoid.
Can we get a zoom on that?
Yes, we can.
This looks like a crinoid.
I agree.
Let's just get a little closer, make sure.
I think it's exactly right. I think that's what that is. I thought crinoids preferred hard substrates. Is that not so? I mean,
To the best of my knowledge, they do, and there's a possibility he might be sitting on a rock
right.
underneath it.
Well, exactly. So it suggests that we're close to rocky outcrops or the base of this slope. This hard
Chris,
sediment
take
may
the
not
cam.
be very far below the soft sediment we're seeing at the surface here.
If it's a swimming crinoid, he's not actually touched
have
the bottom.
control.
He
Go
will
ahead, video.
pick up
Chris.
and swim away, which is what it looks like to me, a cumatulid. So he's not actually attached to the bottom, he's just kind of hanging out. If we were to get close enough to him and disturb him, he would pick up and swim. Very cool.
Fantastic.
That's beautiful, guys. Thank you.
Let's come down a little bit. Look at his base.
Yes.
A little bit
Sometimes these are mistaken for plants
perfect.
by non-biologists. They are definitely not. They are a type of echinoderm. They're related to starfish.
Well, and it's a very old form of life, geologically. I mean, crinoids have been found, what, 300–400 million years ago. So
Yes.
it's a very old form of life, and as I understand it now, they're really only restricted to the deep sea. Is that right?
There are some in the Pacific on the shallow reefs,
Centering
but they
it
look
back up
they
a
look
little.
quite different from this. The stalk crinoids are restricted to the depths,
Okay.
and they've remained almost unchanged from
Right.
such a long time
From the
ago.
Paleozoic,
So
right.
yes.
Hey, Stephanie?
Right.
Yeah, hi Chuck, how are
move
you?
on.
On the
Talk
line
on
we
it,
have
hang on.
Dr. Chuck
Okay.
Messing. He's my master's thesis professor, and he a
about
crinoid
to talk
expert,
about
so
the
feel free to
crinoids.
chime in, sir.
Okay.
Chris, can you reach over and latch my OK?
Well, this is
external
a
piece?
member of the family Atelocrinidae.
A pelacrinidae.
E.
The science
Yeah,
on
you
shore
can
is
tell
describing it.
because
Yeah, I'm listening
when
now.
you zoom
Oh,
inches,
I think Chuck's cutting out.
We're losing you a little, Chuck, on audio.
let's see.
It would be great if you're on the event
In
log.
any case, can you hear me?
You're cutting in and out, so you could just go ahead and start over.
We've gotta
Okay.
move on, Roland.
If you look at, I don't have the visual anymore, but if you look at the base of the arms, you can see there are no little side branches near the base. And at the very tips of the arms end in a very, very long, slender filament. These guys can swim, and they are absolutely unique. I've got the video
Go ahead and
again.
lift off.
They're absolutely unique in their morphology, and
Lifting
it's a strange thing because their most close relatives are
move your
stalked
camera back.
crinoids, whose
Full wide.
closest relatives,
Go ahead, Bridge.
again, are unstalked crinoids.
Thank you so much. That's really neat.
Uh,
Are these
yeah, let
pretty
me check.
rare?
The pilots. Thanks, Bridge.
Well,
Good eye rolling.
they've been trawled fairly frequently
Pilots,
in the Gulf of
Bridge is
Mexico.
requesting
There are a
another
couple of different
5-degree
species.
heading
How
change
deep are
to
you?
report.
We're about 1,150 meters.
Yeah, Bridge, that should be fine.
I just said good
So
eye
that
rolling.
is
you.
probably
Okay, Bobby, you swing
Atelocrinus
to your starboard, just like
helgae.
you're doing.
Telocrine.
I'll keep coming.
Thank
to
you.
my
That is
port.
awesome.
Wonderful. Thank you very much.
Eventually get to 340.
You've just heard a wonderful example of what makes telepresence so unique. We've got a very small group out here, and only a couple of scientists. Got a lot of pilots and a lot of engineers and a lot of crew, but not very many scientists. And yet we've got a vast storehouse of expertise ashore that can connect with us on video and audio to the ship and talk to the wide world in essentially real time. And you just had a great example of that. We had a crinoid expert talking to us about the crinoids we were looking at on the seafloor in real time, and that's a unique aspect of what makes ocean exploration and telepresence such a wonderful activity.
What is this,
Thanks
Roland?
again, Chuck.
Said
And feel free to
shells?
chime in
Think it's
anytime, and
a
that's true of
crab
all of
shell?
you. We're only as
Nope,
good
it's
out
shrimp.
here as the team members ashore
Next to a coral.
help to make us, so thanks very much.
Come wide again. Just real quick.
Looks like we might have a coral ahead. If we could zoom in on that.
Yeah. Chris, again, take camera.
I have control.
All right, let's go in. Right center. Yep.
Looks like we have a small gorgonian with a shrimp.
Sitting on a clamshell.
Yep. And a tiny brittle star, yes. Brian, can you see this video yet?
Yes, just coming through now.
Uh,
Definitely an
Pilots, can we
coral.
get the lasers on, please?
Don't know the species.
Lasers coming on. Oh boy.
Guys, can we get the lasers, please?
Lasers
Yeah,
are on?
lasers are on. You won't be able to see them until we zoom
And
out.
we're too close. We're a little
Yeah.
too close. We've got to come back out a bit
Yeah.
to see the lasers.
We'll do that once we
You
complete
got enough
the
of this
imaging.
rolling?
Oh, there.
Give me a little bit more. There's a couple of enemies in the foreground that I'm trying to focus on.
Your
Very nice, gentlemen.
your bubble cam just reset as well.
Okay.
Mhm. We have an
That
armored
keeps
shrimp
resetting, I
next
don't
to
know
it.
why.
Octocoral.
Also something in the foreground on a stalk.
Little tube worm.
No, it's actually some type of anemone type.
There's a possibility there's a tube worm too in between the sea fan and the shrimp. There we go. We focused on it. Yep, serpulid.
Now, that would be a different tube worm than the kinds of tube worms we were seeing associated with the seeps, though, on
Yeah,
dives 1 and 2,
absolutely.
right? It's a
Absolutely.
very different type of.
All right, rolling.
Mm-hmm.
Good.
Okay, pulling out.
Yeah, if we can zoom out.
Zoom out.
Get the
Yeah, let's
lasers.
get the lasers on that before we leave, though.
Lasers are on.
Also a little hermit crab, although I'm not sure we.
There we go.
Perfect.
For those of
Maybe
you
close-up.
Tilt that
looking at the video
let's
right
tilt
now,
up.
those lasers are 10 centimeters apart, about 4 inches, and it's a primary way in which we can judge scale out here on the videos. On the
Pulling
first dive, they weren't
All right,
working, so it's
lifting
very
off.
useful
Moving on.
to have them. Thank you, gentlemen.
Tiny little guy.
Yeah, he's even pretty small. 10 centimeters wide, about.
Just a quick snap zoom here, rolling. Let's just identify this.
Is that Sargassum detritus
Yep.
I'm seeing?
Okay, let's come wide.
Certainly looks like it. We can turn the lasers off, guys, when we're transiting.
Roger that. Lasers coming off, co-pilot.
Come again, pilot. Lasers off.
Lasers off, please. You ready to make movement?
Sure.
Are
You
you-
have your camera back.
Yes.
Or are you and Todd working on something? there?
Stand by one.
I don't know if he was talking to me or not, but he'll address me.
Is that a white shell or shrimp on the upper right?
Okay, pilot, back with you. I am ready for a move.
Everything okay over there?
Yeah, we just need to make note of the turns into 6-8. Todd and I were talking. We're good to go.
Nav, let's get underway.
We're now essentially at the base of the steeper part of the slope,
Can
and
you
we'll
restart
be
the sonar,
starting to
please?
work up slope, and we'll be looking for hard ground
So
targets.
pilots, what if we start with a
We're
20
presently
meter
still at a
move,
depth of about 1,150
bearing
meters,
toward
but we will be
waypoint
starting
2
to climb
at
here shortly.
0.2 knots?
An enemy
Got
crabs.
one of those crabs with an enemy on its back.
Yeah, call out the heading there, Dan.
Bearing
Can we zoom, pilot?
Yeah, I'm trying not to
340.
take up too much dust because it was a quick stop.
Uh,
Go ahead.
as long as they're not gonna spend time here, that's okay with
You
co-pilot.
yeah, you guys can get going.
Okay.
Your mic's not open, Stephanie.
Bridge
We
now.
have a crab with a resident anemone.
Yeah, I'd like to request a ship move. Let's go
Sorry
20
for the dust
meters
rolling.
at
Yep.
bearing
Yeah, let's get outta
340
here.
Pulling out.
at 0.2
Lifting
knots.
off.
Tried to stop too quickly for this guy.
You got it. Thank you.
Those clouds you see in the D2 view are generally caused by us. As we transit the seafloor, we use thrusters to move D2, and sometimes they kick up a little soft sediment at the surface, and then when we place D2 on the seafloor for an operation, we're not sampling, but when we might take a temperature measurement or so on, that can kick up a good deal of sediment. We try not to do that because video is our primary tool, and we want the water to stay as clear as possible. And one of the skills of being an ROV pilot is to is to do complicated maneuvers with a 4-ton in air piece of equipment without stirring up soft sediment within a meter or so of the ROV. So everyone out there should be aware of and respect the skill involved in doing that. That's why we move slowly. In addition to wanting to examine what we're looking at on the seafloor, it's also to prevent ourselves from disturbing it as we move. This is the Okeanos Explorer.
Ooh. Looks like we're coming on some hard bottom, and we definitely have a type of coral here.
Oh yeah, we'll definitely want to zoom on that.
Yep.
Looks like a
We
black
definitely need to zoom on this, guys.
Roger that.
It looks like a black coral to me with a squat lobster.
And lasers, gentlemen, when you get a chance.
Lasers on.
There's a starfish in the background also.
Yeah, let's go, Roland.
Often see these corals in with organisms
we have
perched in
more
them.
corals.
We've got a shrimp there and a squat lobster.
Squat
I guess.
lobster.
They get up there to get away from the sediment if they're filter feeders. Gives them a little bit better feeling for feeding, I presume.
Beautiful.
Yes, Stephanie?
Yes, because
And,
then they can filter out of the water column.
And that is what we call a black coral, right?
Yes, or antipetherian.
Antipetherian.
There's also another one on the top
Very
left.
healthy specimen of same, I would say.
And there's another one in the top left
Is
corner,
that
and
full zoom,
shrimp.
Roland?
No, it isn't.
They-
Beautiful zoom, guys.
Is beautiful, isn't it? They used to think antipetherians were antipathogenic, which is where they got their name.
Oh, really? And
Uh-huh.
why? And what made them think that?
You know, I don't know,
Huh.
but they're pretty common for the black market for jewelry trade.
Right.
And depending on the species, they can go up to about $30 an ounce, which is quite large, quite high.
I'd be interested in knowing what a specimen like this, how long it takes to reach this size.
Watch lead, any
in this
areas?
spot.
Yeah,
you wanna
not sure the
focus on?
growth rate.
I think we're going to want to zoom out and back in on the smaller organisms on board. We just did the squat
Bridge,
lobster.
co-pilot.
I think I'd get a zoom on the shrimp.
Okay.
I think your mic's open.
There's our shrimp.
Roger that.
A type of Penaeid, or
Copy that,
broken
Nat.
back shrimp.
Oop,
Yes, thank you,
Sorry about
co-pilot.
that video.
And then we'll want to zoom back out and get a good shot of this framed with the lasers.
I think I'm slowly sliding around here, Roland. You having trouble focusing?
Science servers also some small.
Brian, how old would a specimen like this be? Do you have any idea?
Can you pull him out, Roland?
Hard to say. Could be in the hundreds of years.
Uh-huh.
I'd there may be an expert
Oh, sorry.
online in the chat room.
Certainly one of the real revelations to me as a geologist has been how old some of these deep water corals
I'm gonna
are.
come back left to this
And
guy.
and because we are worried about the environment in the Gulf of Mexico relative to commercial development of oil and gas, one of the big issues, and we were talking to members of the Bureau of Ocean Energy Management the last couple of days, is to protect areas like this from commercial
Okay, I'm pulling to
oil
the
and gas
stem.
drilling because of the delicate nature of these specimens and how slowly they grow in the deep water environment.
Yes. We'll show a full screen shot.
Yep. Good.
Beautiful, guys. Thank you.
Tilt down.
Oh, there's
I think
one.
Brendan Rourke is in the event log. He would be the one to comment on the age.
See yellow one in the
Great.
background.
Brendan, if you're online with us,
base.
that would be great.
Over
You
the
know,
base.
it doesn't have to happen right now, but over the course of the day, it would be interesting to have a sense of growth rates and so on.
How are we doing, Chris?
I'm being told that if you guys focus the lasers at the base of this coral, we'll be able to get a better estimate of age. So if you could zoom out and do that when you get a chance, that would be great.
Got a couple more minutes probably.
And
I'm
those
looking
are
down
anemones,
75.
yes, small
Roger that.
Yeah,
anemones.
they
at
look
the
like
base.
small anemones.
What's
All
our
right,
what's
let's
our
come wide.
ship speed now?
Our type of
Let's
zoanthid.
come wide and get the lasers.
It could also be
ship is
diphtheid
currently
coral.
stopped.
Oh,
Currently stopped.
we have some time. Then.
Okay,
There you go.
we have time. I don't think I haven't swung all the way, but
It's okay. You
Keep
can
an eye
come
on it.
over me.
I think those are tiny clavularia
Fabularia.
octocorals. They had
Yep.
these.
Also an
Is that a
orange.
diphtheid?
Jump, get the coral in the back.
Yeah. Do you want to get this
Eric Cortez is
guy on the left side?
maybe bathypathes.
Go in
Bathypathes.
here, Roland.
Okay.
Yeah,
Bathypathes.
let's go ahead and do that.
And then you can see
Yeah,
further
he's a little
further
bit
out
better,
beyond
the
this,
same
there's a
type.
yellowish thing
The
and a
one
few other
earlier.
things we'll want to zoom in on once we're done capturing everything here.
Sure.
Oh, they're smaller squat lobster, I guess, right?
Mm-hmm.
All
Looks like same species,
When you're
just
done, Roland,
small.
let's go
Small,
wide.
small version of same.
Mm-hmm.
Are you happy with this watch lead? This coral?
Yes, we are.
All right, we'll move
I have
on.
a request for the yellow gorgonian behind you.
We'll go get him, and
I
then
guess
the
in front of you.
large sea star in the background.
Mhm.
Looks like we've got hard substrate either right at the seafloor or immediately beneath it here, and that makes sense because we're at the base of this topographic high, this steep slope that we're about to begin to move up. I think we'll start seeing that quite commonly as we begin to move up slope.
How's our light looking, Roland?
If you got a big pool, I would recommend getting as close to it as you can
Okay.
for the macros.
Here we go.
Okay, you ready?
Yeah. Problem is we can't get too close because then we hit our lower limit on the
All right, we're zooming in on the
rotator.
coral.
Chris?
Gorgonian type octocorals on the left, possibly a Chrysogorgia towards the right.
The yellow one, Chrysogorgia?
No. To the right, the pinkish one we saw yesterday. But
Oh, the pink
right
one.
now, this Gorgonian octocoral, I can't identify it more than that right now.
That is maximum
Yes, this
zoom.
is definitely an octocoral.
Do you need to get
It's yellow.
closer signs?
If you can,
Yeah, let's come wide, Roland.
there also appears
And
to be
also
some
pointing to the yellow one to
small
the right.
solitary corals next to it.
I still
And then
have
the
camera control
Chrysogorgia
pilot.
on the right.
Yeah, is that going
It's
to be
pink.
too much? You going to be able to hit that? Tilt down, if you would.
Sure.
Tilt all the way down, actually.
So all the way down.
The yellow gorgonia might be Acanthogorgia.
Cartway's okay?
Yeah.
Hold there. Hold that.
Holding.
There's a suggestion that it's Acanthogorgia.
Mm-hmm.
Is reasonable.
I can rotate your upper swing arms
We've got
down
lasers
to
on
they're,
it.
they're looking out
Go ahead,
more,
right?
more than usual.
If you guys could get a really
Zoom.
good zoom in on the yellow, perfect.
Coming down.
Nothing worse than getting really close and then finding you can't tilt any more down.
Yeah.
10 centimeters.
Should we focus on an isolated patch, Roland?
Yeah, I'm trying to get some, and I'm going to
Here,
adjust the
How's the
background.
light rolling?
Come right to these separate single branches.
Need more light rolling?
Yes.
Is it okay if I,
Yeah, bring your swing arms down, lowers.
I was just gonna tilt the uppers.
That works too.
So we're focusing on the polyps of what appears to be a Chrysogorgia. Excuse me, Acanthogorgia. It's a yellow gorgonian, or octocoral. And they call it an octocoral because there's 8 tentacles on each polyp.
Is that better, Roland?
Yes.
Sorry, that's me.
Brendan Rourke's guesstimate of the age for that black coral that we saw would be possibly a couple hundred of years.
Yeah.
Birth rates radially are
you
in the
just wanna
range of
continue
10 to
to sort of
20
pivot around and
micrometers
look
per
for any
year.
additional
So
organisms.
we didn't get a good look at the base lasers, but his guess would be a couple hundred years.
Couple hundred
Come
years
down
old.
on the camera, Chris.
Coming down.
There's a squat lobster at the base of this guy.
Keep coming?
Yeah, if you keep come down.
Sure.
I think you're scooting forward.
I think that's full down for me, Bobby.
Beautiful
Well, now-
pilots.
He moved. You happy with this, Roland?
Yep.
Okay.
Roland,
Move
come
on.
full wide. Let's see if this is full down.
Yeah.
Okay. That makes me feel better.
Appears to be growing on a broken off piece
Where do
of
you wanna know,
carbonate
pilot?
or shell.
Oh, hey.
Did you say?
Got three of them here.
There's 2 gorgonians here. If you could zoom in on both, that would be awesome. And lasers when you get a chance.
Yeah, let's start with lasers.
Lasers are on.
So
You got tilt up. Yeah, there you go.
We also have a starfish on the top side of the screen.
on the zoom.
Stand by 1 second. All right, we're ready for zoom now.
And a squat lobster at the base of this guy.
Just let me know where you want to pilot.
Yep, this looks like a Chrysogorgia, which we saw yesterday.
Yeah, we'll start here and then
Beautiful.
just come down to the various.
Twitch. Coming down.
And a little squat lobster's arms.
Gorgeous.
This is remarkable, pilots. Thank you.
Happy to do what we can.
So there are, if you can see on the stem, there's some closed polyps.
All right, let's continue to come down.
Coming
Get
down.
the 2.
Most
Yellow
of them are
corals.
open, filter feeding
Keep
actively.
coming down.
There's a Galatheid right in the
There you go. That's
base
what
of
I wanna
this
see.
rock. And then I think another Chrysogorgia. Let's get a good look at it.
You have to rotate right a little bit.
Looks like another Acanthogorgia there.
Acanthogorgia.
That's all I can give you, Roland.
Okay.
The polyps are alternating.
Also have something in the bottom center foreground.
Let's keep
And
coming down.
then, yes, there is something in the very center screen at the base. Oops,
Very, yeah.
right there.
Translucent, delicate.
Yes, I'm not sure what it is. It looks like an Adarian of some kind. I honestly have no idea what to call that.
Okay, pulling out.
Me neither.
Oh, Scott France says it might be a very young
Trying to think it's a
black
very
coral.
young black coral.
Yeah.
Oh,
I've never seen that before. Very cool.
got a few hundred years to go.
Wide.
Nope.
Well, that's
Yes.
great if it is. I mean, it
Want
would
to try getting
show
that starfish from
signs
here?
of recruitment.
Since I'm
And
sitting
there's
down.
a fish under the
Yeah, it's
rock.
very important
Yeah,
for the
we won't get
habitat.
It's important.
macro, but we'll get a pretty good picture.
I think we're going to go for the starfish
Sea
next.
star, rather.
That is a type of gawny asteroid.
And we've got a fish off to the right of it too, in that under that overhang.
Just a few more corals.
And the yellow, yellow coral on the right.
I'll get a 3-shot.
Yes, it looks to me it looks like it is feeding. Starfish
little
feed
tilt
by
up.
extruding their stomachs
There we go.
out of their bodies and enveloping the food and dissolving it. And they usually will get in this position where they puff up in the center. Really the only way to tell would be to flip them over.
That fish is very busy. It looks like he's doing some excavating himself.
Or hiding from our lights.
Or hiding. Yeah, that's right.
Are you happy with this area, Watch Lead? Shall we continue?
Yep.
I think we are. I think we can
Stand by.
proceed.
Is that a cup coral on the right?
Pilots, how do
Yeah,
feel
before you
about
run off, there's a small cup coral on the right in the rock.
Is the view from here okay, Watch Lead, or would you like to get closer to it?
It's fine. Can you zoom in
Roland, let's get a zoom.
without
from here.
moving?
Yeah.
Beautiful. We have a definite cup coral.
Is that full zoom?
Nope. Close though.
Yeah, I'm pretty far
That's
away.
maximum.
We can get closer, Watch Lead, if you'd like to, but.
You guys are fine. Thank
Okay.
you.
All right, let's come wide and.
And then,
Move
seems
on.
to be a shell or a gastropod shell underneath them.
You can
Thanks,
see
guys.
see the difference in the seafloor now as we approach the base of this steeper part of this slope. More small cobbles, more habitat, hard substrate for the biology we expect to see as we begin to climb
So
this slope.
pilots, how about another
Found a lot of life in that
20
little
meter
area.
move towards bearing 345 toward waypoint 2 at 0.1 knots?
Yeah, why don't you get out there a little bit, Bobby? I'm still looking at you at 50.
Roger, let me just swim over this and
Sure.
I'll continue on. Rolling real quick snap zoom.
Yeah, stand by nav. As soon as he
Hold
gets
there.
out a little bit further, since we're going uphill, we'll call that in.
Other Chrysogorgia.
that.
Oh, you're going to get us a pirouette video. And not a great one at that. Moving on.
With associates,
I'm sorry, Brian, you cut out. What'd you say?
I said with associates.
Yes.
Looks like there's a shrimp in there at least.
I think there's a shrimp in every single one of them.
We've-
Without fail.
2 years ago out here, we almost every deep coral we
Still
saw
fun
had a
to play.
brittle star wrapped around it. I haven't seen that so much yet, although we may do that. Shrimp and brittle stars.
There's another Chrysogorgia on the rocks.
It's the
Lots of marine snow. It's block the
Yeah.
water column.
Yeah,
Usually a good sign.
certainly appears to be more today than yesterday and more yesterday than the day before. I guess we're in the middle of spring blooms and things out here, yes? Those of you who are biologists, I would expect this to be a peak time for phytoplankton and associated zooplankton to be in the water column.
Hold on, let's go in here.
Yeah, if we can get a quick zoom in here on this little patch, I'd like to see so the Chrysogorgia and the red thing to the left.
Yep. You're on a bit of delay. They're zooming in on the pink.
Soft coral?
Yes, right now.
Stable for
Maybe
more.
not Anthomastus. Scott France agrees.
Yeah, let's go more.
Anthomastus.
See, we saw a lot of those in the Canyons expedition last year, northeast U.S. in the Atlantic. True soft coral, no skeleton.
There's a brittle star right
You can see he's
below it.
clear on the stalks.
Clear on this.
Let's come down a little bit.
We have a small sponge and brittle star arms coming up through the rock.
Can't tell, but is there a very small, possibly dead Gorgonian off to the left, the yellowish, stalky thing?
Guys, could you shoot to the left a little bit? We saw a stalk we're going to look at.
It's okay. I think we got it. We got it off to the corner of the shot.
There's this small serpulid worm on the bottom right.
Yeah, I saw
Back
that.
at it later.
Go back to
I
him.
do not think he's dead. I think he's got a couple of polyps on him because we're back.
There's maximum
We're
zoom.
back on him
Let's
now.
come wide.
And his resident shrimp.
Oh, yeah.
Let's see if we can do this.
Oh, you can get a shrimp.
Seat pin in the center of the group.
There's two Chrysogorgias here.
The ship store is open. The ship store is open.
The ship store's open. Excuse me, guys. I'm going to go buy a shirt. Parentiopathes, possibly, we're being
All right, let's
told.
go back to the left a little.
Yes. Perfect.
Just to the left of the Chrysogorgia, Scott. Oh, yeah, there we go. Good
Yeah.
eye. Scott
Drifting.
France
So,
on event log.
so there's a black stalked coral right in front of you on the upper right. If you could zoom in on him.
If we get a zoom on that, yeah,
Okay, let's go
the
in.
black coral.
Video.
Yeah.
They were asking
Oh,
for
no.
the one on the upper right.
Yeah, I just want to get him one more shot of this one.
Not that guy.
Yep.
Okay.
Stand by one, watch the. You're full down. Okay.
Yes.
Another good Anthomastus shot.
Here's the worm.
I'm sorry, Brian, what did you say?
I said, sorry, you guys having trouble hearing me?
I am a little bit, but I can turn you up. Let's see.
You're breaking up a bit, Brian. So slow and steady, and we'll try to hear you.
That's full down.
I was saying if we could get a zoom on the black coral, the stalked black coral that was to the left of the Chrysogorgia, that would be helpful.
Yeah, we have a pace of zooming in. We like to hold these zooms for a bit to get close-up video, and that's modulated not just by us, but by the pilots working with video on board. So there's a pace here, and we like to do it in a deliberate way. So
Is that full zoom video?
no worries.
That is. Well,
Okay.
I pulled out a little bit to
Sure.
get
Great.
the shrimp. You want to go ahead and go up to that?
Yeah, let's
black-
scroll
Sure.
up. Our camera can't tilt far enough down to get a good visual of the worm. So let's come up, hold that zoom. Let's go on the
Holding.
black coral at the top of the screen.
All right, we're going to the black coral next.
Pushing in. Going for macro.
Where do you want to start?
Parentiopathes is a suggestion from shoreside.
Bringing it back, sorry.
Very cool.
Okay, pulling out for a full shot.
Now that one doesn't look as healthy as the one we just looked at. Is that an accurate observation on my part?
Not necessarily. It might be a species difference, but if someone on the line or on the...
Scott France just commented that, yes, it's not in great shape.
Okay, it is not in great shape, yes.
Actually, center. You see it just before the big coral.
In the mud?
The rock to the right. Right, coming into the bottom screen.
Okay.
Really shows a polyp swell against the dark background.
It's a zigzag stalk. It's very small.
Okay. You just continue to rotate and go up the other stalk, and I'll follow your focus.
Let's come up.
We also have small white sponges. And our Chrysogorgia with a resident shrimp.
I keep coming up.
I'm coming.
And our squat lobster.
Want to get the squat lobster full?
All right, when you're happy, video.
Let's get a full.
Are seeing disarticulated bivalve shells around. Bob Carney's mentioned on previous dives that the scattering of these shells, which was much more common near the seeps we've been looking at the last couple of days, were related to predation. We are seeing some of those shells here that may have ended up on the bottom of the slope from higher up.
Okay.
So we want to be on the lookout for those as well.
Any other requests in this area? Shoreside?
No, I think we're good.
Okay. Thank you, pilots. We can move on. Beautiful job, by the way.
Thank you.
Okay, Nav, back with you.
Yep, are we ready for that ship move?
I believe so, as long as pilot is.
So I'm looking at a move of...
Oh.
Sorry, Nav. We keep telling you to go, and then we.
So looking at a ship move of 20 meters at bearing 345, 0.1 knots.
We have a small patch of hard bottom in front of us.
That works for me.
Nope.
And as we get closer, I see something growing on it.
Pilot, I'm at 335.
Some fan of some type, I think.
Roger that. Thanks, co-pilot.
Getting close to the steeper part of this slope now. As we have said, the operational tempo here is slow. Our goal on this dive is to examine biological habitats as we climb the slope. So of necessity, this is going to be a slow operational tempo. We're zooming in on specimens of corals and related organisms while we move up. Got another scattered carbonate, looks to be a carbonate hard ground, similar to what we've seen on previous dives. We're on the topographic high west of the oily seepy high that we were on yesterday.
Anything of interest
we
here?
don't have any evidence for seeps in this area, but from a geological perspective, it's not very different from what we were on yesterday.
Watch leads. Anything of interest here?
I'm sorry, could you please repeat?
Anything of interest here in particular?
Yeah, there's 4 or 5 things if you want to settle down.
Bridge, Nav.
I've got a white sponge at the top.
A ship move,
and a yellow
20
gorgonian.
meters at bearing 345 at 0.1 knots. That's it. Go ahead.
Can you take control of my camera, co-pilot?
Looks like we have 3, 4.
Which direction do you want?
Just stand by for me.
Okay.
Come down.
Okay,
actually.
great. Thank
3
you.
or 4 or 5 yellow
Thank you.
octocorals as well as a couple of pink ones.
Let's check this coral here first before I go.
Octocorals as well as a couple of pink ones.
Getting a lot of background noise.
Yeah, we're getting a lot of feedback. If you guys could mute your phone on the line, please.
I'm coming up. Just stand by, co-pilot. Just background. Am I at the end of the tether?
You're getting close. I can come down a little and give you a little slack.
John, is that you?
I just need to let all over.
Yes, hi. This is John Reed.
Hi, John. How are you doing? For everybody out there, this is John Reed. He's a deep sea coral reef biologist and my boss. And he is calling, I'm assuming, to help identify some corals.
Nav, you said you were negative.
If you could go ahead and pick your phone up, that would be great. Go ahead.
Yeah, it was pushing me ever so slightly
You knew it all.
down into the
I
mud.
don't know it all, for
so you
sure.
have to give it some effort to get up, otherwise you're just dragging your toes.
You just tell them you know it all.
I pretend like I do.
Kind of drag along the mud.
Chuck was on the line earlier. He identified a crinoid for us.
Mm-hmm.
That was pretty fun. Are you watching in the ECC at Harbor Branch?
Yes, I am.
So you're getting a really good live feed?
Yeah, it looks beautiful. We got 6 students here.
Oh, wonderful. So feel free to chime in and identify some of these corals for us.
John,
Workshop.
Jamie Austin, Stephanie's doing a great job out here.
Thank
you. Great. I knew she would.
Come on in, video.
I see a sponge in the upper part of this picture. I'm sure you can ID
Workshop
for me.
ROV nav.
So right now we're zooming in on a yellow gorgonian.
That's a partial.
Sorry, go ahead.
I was told to give reports to the weather to the winch.
You can keep coming.
John, any
Is that
idea
your
what it
call?
is?
Or is there a winch on deck A?
I'll let you know.
Okay.
Looks like another Acanthogorgia.
I was told we were getting
Acanthogorgia.
weather updates to winch.
According to Scott France.
Is winch on ROV workshop?
We saw a couple of
or
these
deck
earlier.
Just tilt down.
Nav, can you release EXRV
Well,
in those conversations?
waves
And a
have
squat
increased
lobster.
4 to 6 feet.
There's also an anemone tucked underneath the
Wind
rock
20
behind
knots.
him. I can't tell the species because he's too far in. We also have some brittle star legs sticking out of the rocks.
Yeah.
Okay, if you're ready, we can go look at that sponge watch lead.
I would love to see that sponge. Thank you.
Sure.
We have a armored shrimp.
What is the width of the lasers?
Our lasers are 10 centimeters.
All right, co-pilot. I'm gonna try to pirouette over there. I don't know if you saw our port verts off.
I see.
I believe we have a hexactinellid
right
up
now.
ahead of us. And we're going to go zoom in on it now.
John, I gather you have students with you at Harbor Branch. Feel free to have the students get on the event log and let us know if they've got any questions or chime in on audio.
How's that video?
One of the great audiences we have are students of varying ages, so welcome to all of you, and we're glad you could join us.
Absolutely. You can jump up
Okay,
here.
tilt up for me, just a little bit. There you go.
Now, they
Thank
really-
you. You can hold there.
That feed is going out to
Try
other
to
people.
do it.
And
John, can you mute the phone, please?
Just gonna do a zoom from here.
you guys.
Tilt
Are we
up.
trying to get the...
Yeah, the white sponge. Tilt up and go
It's
in, roll.
coming.
Ooh.
I can try to rotate a little bit.
Focus in on that. So this looks like a glass sponge.
Look like glass sponges to me.
Hexactinellids.
I'm looking down at you, 40 degrees. and we're
I
10
am not
degrees
sure
off
this
the move.
genus, though. And then there's pink
That's
Ophiuroids.
a partial.
Okay.
Looks like a type of
Come-
Ophiothrix.
Keep coming.
based on its spininess.
So a hexactinellid would be the family? Is that right?
Hexactinellid is the class.
Would that be the
Maximum?
class? Okay.
Yes. So phylum Porifera.
This is hexactinellid. There's several genera. It actually could be the one we see typically at this depth, like Euplectella. There's several species and even genera that I wouldn't want to guess at right now.
Okay.
But if we could
Go ahead,
zoom
pilot.
in,
Hey, you have control of
look
my
at the
main HD
pattern.
on the right there.
Okay.
If you would.
We'd like to get a closer zoom on these glass sponges directly in front of us, guys, if possible.
To identify it from that.
Maximum, you want me to
Both on
pull
the
out so
inside
you move
and
in?
the outside
Yeah,
of
if
the
you would, just
sponge.
come full
You
wide.
can
I'll.
Full
zoom.
wide.
What are you trying to?
Okay, they're
focus on,
gonna
pilot?
probably have to pull out,
Just
scoot
stand
forward,
by
and then
now.
re-zoom in.
I'm gonna come.
So let's
come
they're gonna reposition and get a closer look.
You might explain to everybody, Steph, why we call it a glass sponge.
It's silicious, right? It's made from silicate. So instead of a typical demo sponge, which is spicules, I'm not sure what demo sponges are made from. Calcium carbonate? No. John, do you know that?
Yes, they would be.
They are calcium. Oh, yes, demo sponges are silicious. Okay.
These are
Start to come in.
these are pure silica, right?
Actually, come back out.
That's what I thought.
I'm being pulled.
But demo sponges are as well.
Pilot, just a heads up. I'm looking down
So
at you at 50
when we're done
degrees.
with the
Okay, are
sponge,
we
let's
moving?
just march around. There's
Okay.
and then there's the black coral behind that, and then over to the far right there. Like, and we'll want to get zooms on all those.
Sure, no
Ship
problem.
should be coming to a stop soon,
Okay.
though. It was a short move.
Copy that. Thanks, now.
Go bridge. Roger that. Thank you.
You know, geology is not the focus
All right,
here,
Roland,
but
come in.
the
Co-pilot,
nature
I'm
of
gonna
this
need you here in a
outcrop is
second.
quite interesting as well. You're seeing cobbles of varying
Video feed
shapes
just
and
went down.
sizes. Some of them even look tube-like, and
Co-pilot,
that can be the
pilot.
nature of orthogenic or what we call
I need you
formed
to
in
go ahead,
place
camera.
rocks. These rocks were formed beneath the surface of the sediment and then have been exhumed or
We're focusing
excavated
on the white
by currents
sponge.
and so on. And orthogenic rocks are formed in pore spaces and cracks and so on from percolating fluids,
Hey, Dan, I'm here as well.
which are
So
here in abundance in the Gulf of
we
Mexico.
need you on
so
headset, so if you have
gives
questions,
these
feel
odd
free
shapes
to
and
send them my
sizes of
way.
rocks that we're looking at.
Copy that. Thank you.
So the goal here is to focus on the inside and outside of the sponge to get a pattern so we can identify it.
Is that full, Roland?
That is
That's great.
maximum.
Okay. How's that watch lead?
Perfect, guys. Thanks.
Ship is stopped.
Copy that. Thanks, now.
Looks like there's a zoanthid too on the rock to the left. So you can see the pattern.
I would put my guess at iophyton. Start with that. I-O-P-H-I-T-O-N.
Phyophyte?
Internal word. Iophyton, I think. I-O-P-H-I.
Okay. T-I-O-N.
O-T-O-N.
O-T-O-N.
Iophyton.
Phyophyton.
Hard to image now, Roland.
Very
Thank you
difficult.
so much,
Right now,
John.
focused in the center
You guys
of the
can go ahead
back.
and look at the coral
It's
snakes.
always
Thank
nice
you.
to have a boss
If I try to
online
focus on the-
Which corals,
keep,
watch lead?
to keep the team honest.
Watch lead, which corals?
That's right.
Go ahead and zoom out. There's a bunch of stock
Okay.
stuff in the back.
Come out, Roland.
Pilot, my Sirius cam is at about 65 degrees looking down at you right now.
Okay, thanks.
Full wide.
All right, watch lead.
All right, so
I
there's
think we can
there's
skip over the Acanthogorgia and go to the black coral.
the black coral in the back. Was the request?
Yeah.
Okay.
You see it right in front of us with the crab on it?
Yeah, let's
Co-pilot, as you swing over
try to move over there.
the ROV,
Full wide, Roland.
pull your altitude up a little
You
bit.
are?
Copy that, thanks.
And that little white mat that we saw to the left of the sponge was actually an octocoral as well.
Oh, nice.
Octocoral. Scott France is saying.
Like it was encrusting.
Similar to the clavulare, the purple we saw earlier.
Okay.
Similar growth form.
Thank you, Roland.
All right, video start to come in.
Pushing in.
And we've got our squat lobster again. This looks
Axel.
like the same species we saw earlier.
Copy that. Do we want to get a good look at the base here, watch lead?
Yeah, if you could laser the base, that would be great.
Okay. Yeah, I'll tell you when to tilt down. Roland, sorry. That's good right there. Hold there. Let us know when you're done, Roland.
Yep. We want to get tight on the base, centered.
Copy that. Go ahead, Dan.
And then we'll
Possibly
want to
this is a
pull out and laser
bathypathes.
it.
Oh, not a bathypathes. There's a debate on our chat room. Earlier, I think we called this parantipathes.
That's probably correct.
Okay, if we want to laser the base.
Sure, you might have to tilt up a fair amount, Dan. Yeah, there you go.
There we go. We're lasering the base of this. Innovatherian.
Hi, everybody. Your squat lobster belongs to the family Chirostylidae.
Chirostylidae.
If you can get a sharp picture of it, I can identify it later.
If
Wonderful.
You can get one more image of the squat lobster.
Sure thing. Coming up, co-pilot.
I will ask the pilots. I have a crab expert on the line who would like a very good close-up and still of the squat lobster.
All right. See what we can do. Come down.
Do you need specific mouth parts, or what would you like me to focus in on?
The area around the eyes.
If you could focus in around the eyes, that would be awesome.
It's maximum zoom.
That might be the best we get. Watch these. I can try to pick up and sit back down.
Typically, this has been good enough.
Okay. If we could get a still of that.
Okay, let's just fill all of his legs in the frame.
Sure.
Bandages,
Was that sufficient?
That should be great.
Okay. That's great, guys. You can go ahead and move on to the next coral.
Copy that. If you, when you're done rolling, come full wide.
Okay. Can we get
Brian, which one did you have your eye on next?
Of the entire coral.
The white one all the way around to the
So right there.
sort of a fan
Tilt
shape.
down.
Come on down, Dan.
Okay, hold that. Just get this for about several seconds. We can lose the lasers.
Go ahead, Dan. Turn the lasers off. Should be in bank 1. ROV.
If you pan far right around to the other side of this shell mound.
Yeah, I'm waiting for them to pull the camera back so that I can see exactly
We're
where
going to pull out wide. We're going to switch out video engineers.
So there, you said there's a white one. I see it on the back.
You're full wide.
Back right.
Copy that. Thanks.
The next car we'd like to take a look at.
The white in the top right of the screen, watch lead. White coral.
You are correct. Thank you.
Okay, pilots, going to be with you just a little bit longer.
Okay. Nav, pilot. You think clockwise or all the way back around? Let's go.
I believe it was upper right.
Let's go counterclockwise. What do you think?
To get to the white one?
Yeah.
I'd go counterclockwise.
Yeah.
I'd even more maybe come around and point towards Sirius, because look at where the current's going.
Yeah.
So if you put your tail to the right, you're going to blow into your coral.
Right. Okay.
So
Okay, video's changing out.
Kind of see where your mud track is. I'd probably land in
right.
even more to the left of that. Back with you, survey 1. Speed things up. Oh, okay.
And Brian, your coral model is based on presence absence, not necessarily percent cover, correct?
Correct.
Roger
actually,
that.
it's only
I
based
had
on
not
presence.
heard any
Presence.
So it's a presence
clarity
only
on
model.
how important
You
the
don't
top
have
was or
reliable
not. I
absence
had heard that
Right.
we got plenty of
So,
time, move slow
It's the
a few
minimum
times. So I'll
possible
check in
amount
with
of information
Watchley.
you can have and still create a model. And actually, one of the reasons we're interested in field expeditions like this is,
You got it.
and in also review of
I
older
hear ya.
video past OER expeditions, is to try to, over the next few years, build up a database of absences,
Okay.
develop an improved model.
I have you, Ed.
But yeah, this is a presence-only model that predicts the
Copy that,
likelihood
thanks.
of suitable habitat.
As soon as this
in the area
clears,
based
we're going to try
on
to get a shot of
a whole range
this
of
white
environmental
coral.
conditions.
Center screen.
And it does not predict the abundance or the diversity.
Okay.
So even presence could just mean
Dan,
a
if you
few
could tilt
scattered
down just a
individuals
little bit. I'm going to have to
that are
rotate.
very
But
small, or it could mean a coral garden with high diversity and high density.
Okay.
We don't find out until we get down here.
Will that do,
Yeah.
pilot?
yeah. If you want to start coming in, Ed, I know I'm going to have to rotate, but
All right, we're zooming in on our white coral. My guess would be Carihoa, but that is a wild one.
yeah, go for
I'm
it.
sure if John is listening, he can chime in and correct me.
Tilt down a little, Dan.
We're hoping Scott France can take a look at this one.
You can keep coming as much as you got, Ed.
Beautiful. Beautiful, guys. Thank you.
Scott says Denoctocoral and a Primnoid
Primnoid, okay.
coral.
Do you want to get a shot of the base watch lead?
Yeah, if you could scan the whole thing,
Okay.
that'd be great.
Let me know when, pilot.
Yeah, Ed, as long as you're okay, we're going to tilt down. Go ahead, Dan.
Oh, little anemone at the base.
Let us know when you'd like lasers on, watch lead.
Sure. Perfect.
That little orange-red shrimp to your left is probably a thread-leg shrimp, a genus called Nematocarcinus. And you had another shrimp, a pink one on the coral itself,
How's
which
that
is probably
video?
Bathypilmonella.
All right.
Wonderful. Thank you so much. Okay, Brian, since this is your dive, we can either go super slow like we have been going, or we can make it up to the next
Nice video.
waypoint and try and get to the top of this hill. What would you like
I
to
think
do?
we should move on. I think we've characterized this little spot.
Move on. Okay. Perfect.
Survey
Thank
1.
you.
I will be keeping an eye on the chat room, though, for any other requests, but I think we can move on.
Okay.
Yeah, to support what you were saying, we have 450 meters to waypoint 2.
Okay.
If we continue at this rate, about 0.1 knot,
And Andrea,
it's going to take
Quattrini in the chat room
146
thinks
minutes,
it's,
over
the
2 hours.
NOAA that we just imaged is likely new for them. For
That's
folks
not
who are
stopping
working
at all, correct?
Hang on, Brian. I'm trying to talk to the navigators. Give me 1 second.
We have gone about 50 meters.
Could you please repeat that? I'm sorry.
Yeah, at this current rate, which is less than 0.1.
Andrea Quattrini, who's in our chat room, chimed in to say that the white from NOAA that we just imaged was likely new observation for the Gulf of Mexico, at least as far as she knows. And she's done quite a bit of work.
Did she know the name of it?
Pilots ready for another move?
Yes.
Yeah, so Brian, we've about 450 meters to the next target. So they were saying if we go at this pace, it'll be a couple hours before we get there. So we are going to be picking up moving. Go ahead.
Yes, ship move, please. 25 meters, bearing 345, 0.1 knots.
Would you agree?
I'm not exactly sure what he was saying because I had like 3 people talking to me at the same
Good
time.
copy.
So I think he said 2 hours to waypoint 2.
So
am I correct?
Pilot, I'm
yes, I'm correct. 2 hours to waypoint 2.
Serious angle, but
approximately 3 hours.
the camera you're looking at right now
so we're
is
about
looking
right
a little
then.
bit port
Good.
of our heading.
Perfect.
Okay. I'll adjust.
Great
our
job
heading
so far,
is,
guys.
remind me of our heading.
345.
345. Copy that. Coming up.
We have an echinoderm on the rock.
Partial zoom, Ed.
Another Crathogorgia.
Just try to. Yeah. That's good, Dan. I just wanted to check it out briefly. Okay, come full wide.
What is that?
Thanks, video. Anything here, watch leads? All right.
I have a couple of Chrysogorgia ahead of us.
Yeah, Nav, waypoints A and D, double checked. You might want to check the conversion again,
yes, I
but
can.
they didn't
How are
look
you?
anywhere near where we want to be. B and C do make a line down the back side of the mound that we're going up, so we'll probably just follow that.
Perfect, and I can't even hear myself, so that's awesome. Thank you.
Great, appreciate it.
Well,
Another Gorgonian. I think that might have been the same one.
I'm getting to the end of my tether. Oh, no, I'm not.
Position is
That's okay.
27046949 by 9248 9722.
Is ship underway, Nav? Nav, is the ship underway? Copy that.
I'm sorry, Brian, did you have a question or did you ask me something?
It's okay. There was snap zoom I was asking about, but I think we moved past
You're
it.
getting a little far to the end of your leash.
Okay. Yeah, I'm not touching anything, and I'm lateraling starboard. That's the port vert. I mean, the starboard vert. Okay. Yeah, I'm getting out there, huh?
We're at a depth of 1,142 meters.
For those of you out there, we are sending you out a quad feed, a split screen feed that has a number of panels on it. We've got our D2 view, our deep discoverer view, our near seafloor ROV view, the Sirius camera sled view. We're sending out HiPAC, our navigation view as well, which we zoom in and out to navigate the vehicles. And we've got one other view,
Dan,
which I believe is the pilot screen,
if you don't mind.
Sure.
which shows attitudes of the vehicles, elevations of the vehicles off the seafloor, and so on.
What are we looking at?
Please chime in and let us know if there are changes in that quad feed that you would like. We want what we're sending out to be as edifying to the larger audience as possible. So feel free to let us know if that's acceptable or not, and we will modify accordingly. This is the Okeanos Explorer.
See anything on sonar?
Looks pretty flat.
I might be able to reach this cluster of things.
If we could just slow down and skim these a little better. Looks like we have at least one Chrysogorgia.
Let's take a zoom on this this coral dead in front of us right now, gentlemen.
We're getting stable.
Yeah, just let me know when you're ready for moving the camera, pilot.
Go ahead and tilt down. Is that full down?
Not quite.
Okay. Yeah. Affirmative nav. Sorry, co-pilot, I'm having trouble here with the...
Take your time.
With no vert. All right, let's try this again. Okay, come on in, Ed.
Yep. I see Chrysogorgia, at least 3 Chrysogorgia.
Copy. Coming up, Dan.
There seems to be another yellow flat fan behind the second Chrysogorgia in the top right.
You can keep coming,
as well as
Ed,
a
if you
small
got it.
pink Gorgonian in the top center. And we have another squat lobster. And there seems to be a yellow one. If you guys could focus in the background of this, there's a yellow Gorgonian in the back.
Okay.
Perfect.
Looks like likely another Acanthogorgia.
Acanthogorgia, okay. The yellow one.
Hey folks, that squat lobster in the foreground
So
is
the
a different
ship's still moving now?
species. It's another chirostylid, and it belongs to a species called Eurotychus nitidus.
Oh my goodness.
Copy that.
Tychus. Wonderful, thank you.
UROPTYCHUSNITIDUS.
Thank you.
All right. Copy that video. Come full wide. What else here, watch lead?
The yellow Gorgonian in the
Middle right?
Yeah,
sort of?
middle right.
Gotcha.
And then a quick scan of the Chrysogorgia at the top, the pink top, and then we can head up.
See what we can do here.
So Scott France is telling us that there are at least 7 species of Chrysogorgia known from the deep Gulf of Mexico, and at least 4 of those are known from the depth range we were seeing today.
So we could be looking at.
We could be seeing multiple species.
Yeah, 1 of 4. It's difficult to tell with Gorgonians. You either have to pull them out and take a sample,
Okay, Dan,
and look at their spicules.
Come in, video.
Another thing you can identify them with is a branching pattern, but for the most part, they're pretty difficult to ID from imagery. Same goes for sponges.
Come back up.
So this is interesting, this yellow one.
Perfect. And feel free to zoom.
I think we called this Acanthogorgia earlier, if it is the same.
Go ahead, Nav. I did had to make a correction on waypoints A and D.
Acanthogorgia.
I wanted to double check that
that's what this is.
my calculations are right, and so I had dropped a waypoint in the meantime, and the other log
Brian,
file
are you
didn't
happy
save
with
that,
this
so
area?
will that work?
I am. Yep, we can press on.
Okay, guys, we can head out. Thank you.
Okay.
Beautiful imagery, by the way.
Come wide video. Copy that.
So we have 3
Roger, 3,
Chrysogorgias
4,
and
5.
1 Acanthogorgia in this little area.
What's your altitude, Dan?
About 17.
Presently at a depth
meters.
of 1,141 meters,
Okay.
and we are getting very
I'm
close
about 14
to the
meters
steeper
above
part
you,
of the slope
looking
here
down
that
at
we're
37
going
degrees.
to be transecting up during
Roger,
the rest
thanks.
of this dive. Still looking at a
Yes,
soft
another ship
sedimented
move, please.
seafloor with scattered
30 meters,
cobbles
bearing
of what are almost
345,
certainly
0.1
autogenic
knots.
or chemically precipitated carbonates. They form the substrate on
Good
which
copy.
the
Take my
corals
camera back.
and related biology that we've been looking at
Got it.
attach themselves to the seafloor on. We can anticipate we're going to
need
see
to,
more and more of this hard substrate as we get closer to the steep part of the slope, and I think we're about to get there. This is the Okeanos Explorer.
Okay. Do we want to look at this coral here, watch leads?
Yeah, it looks like an Anipitherian. So we can zoom in real quick. Again, lasers at the base, please.
Go, co-pilot. Lasers.
Lasers on.
Get the base briefly, and then I might have dusted it up.
This
Yeah.
looks
Yeah,
like the
zoom.
same
I'll take care of your
species, but
camera.
may not be.
Camera up.
The whole fast looks not as round. It's still a branching purple. Black coral.
Yeah, there you go. We got lasers. If you're good with the full frame, yeah, go ahead, Ed.
And he has his resident squat lobster.
It's hard to keep stable.
That's another chirostylid, and it's not Eurotychus. And whatever it is, as far as I know, there are no accurate records of its host.
Would you like us to zoom in on him? Maybe you can get a better idea?
Yes, please, because it might be worth a note to figure out what it lives on.
Okay, hang on 1 second. Hey guys, I have a request to zoom in on the face of that crab that's on the coral. Thank you.
Let's see what we can do. It's a little tricky here. Let's get back in. Keep my camera, Dan.
You got it.
There is another black coral in the background.
All right, come on in, video. We're aiming for the squat lobster, I believe, and come down.
So who keeps requesting me for crab identifications, or telling me crab identifications? Can you tell me what your name is?
Yeah.
Hey, Stephanie, that's Mary Wickston up at Texas A&M University.
Wonderful, thank you.
Come back down. That was a twitch.
So Mary, is this good for you? Are you guys getting a good shot of him?
It's a lovely picture. Whoop, get him up a bit.
Yeah. I think you've got a little bit of a delay.
There he is.
Yeah, perfect.
And Mary, you have about a 10-second delay off their video. So.
How's that, watch leads?
See that moving
That is
camera
perfect,
about 10
guys. I
seconds
think
later
we're ready to move on. Thank you so much.
Scott France is suggesting this coral, his best guess is that this coral may be in the
I think there's another coral behind and to the right of this, this coral they want to take a look at.
Roger that, watch
I'm sorry, Brian. Could you repeat that?
There's been a debate going on in the chat room about the ID for these black corals we've been seeing. Scott France's best guess is that this is a black coral in the genus Cybopathes.
Camera, Dan, I'm sure.
Not Parentopathes, not Bathypathes, as we had suggested before.
Okay.
Yeah, it looks like we've got two corals there we're going to want to take a look at, one in the left foreground and one in the background.
Copy that.
Yep.
There's been some evidence of these various
move the serious cam
particular
5 degrees to port. Just keep an eye on you.
branched gorgonians or anthopterans, but in many cases we don't know what they prefer. Eurotychus prefers to live on Chrysogorgia, but as for this other one, it looks like it's hanging out in anthopterans.
Very neat. And this is all new? Totally unknown.
Okay, come down, Dan.
Most of the records of these things come from trawls or dredges. You had a beat-up specimen that came from who knew what.
Right, so this in situ imagery is actually really beneficial for identifying behavior.
It certainly is.
Trying to get the end of the claw.
Yeah, I don't think that particular organism would be too happy once it got to the surface in a trawl.
Once it got trawled at all.
Broken into shreds.
Yeah. Really identifying them from body parts at that point.
You also want to let us know, everyone, if we're getting you, we can get you a zoom, but if we're not in the right orientation for the zoom you want, let us know.
Can we get lasers on, please?
Sure.
You got it.
How's this watch lead with this particular coral?
Is everyone happy with the coral? Can we move on to the next one?
Sure. Come,
yep.
Yep, we're good. Go ahead to the next one. Thank you.
Come a little wide, video. I think we can just scoot over to it. That's good, right
There's
there.
another crab at the base of that. Here's this another yellow gorgonian right in front of us.
Again, we're also seeing pretty ubiquitous scattered bivalves. They're sedimented, so they're not fresh on the seafloor, but there are a lot of them. Bivalve shelves, I should say, not live bivalves.
Oh, we've got another crinoid on him.
Yeah,
Chuck, if you're listening, feel free to ID.
Video. Let me center it for you.
Yep. We have a cumatulid crinoid sitting on another, this yellow gorgonian.
Perfect. sorry, Ed.
And this is the swimming crinoid, right? This is the one
Yes.
that moves. So
Correct.
he's just up there to get a little bit better filter feeding
Come down
position.
for me, Dan.
Yes.
That's great.
Correct.
That's great.
Okay, we're more stable there, Ed. If you want to keep coming in. Sure. And then,
Wonderful, guys. Thank you.
Another Acanthogorgia.
Come down,
Yes,
Dan.
Acanthogorgia.
Everyone out there,
I might be maxed out.
All the way down?
We are in the middle of
Yeah.
dive
Okay.
3 of Expedition
Come a little
1402,
wide video.
Leg 3 of the Okeanos Explorer
See if I can
sojourn to the Gulf of Mexico this year.
maybe
We're in a
back
water
up.
depth of about 1,140 meters, and we're looking at a series of deep water coral
Yeah, let's see if I can back up
habitats,
a little bit.
looking at symbionts associated with those corals on small carbonate hard grounds, orthogenic carbonates. We're at the base of a steep slope
Ah,
of a
come wide.
topographic high in the north central Gulf of Mexico. And
Thanks,
we'll be
video.
climbing about 100 meters to the top of this feature, and then if we have time, looking around
Yeah,
Want me to
come-
pull up?
yeah.
for additional habitat
Please.
for these organisms
All right.
at the top of the feature. This is the Okeanos Explorer.
Okay, I'm gonna
Can we
correct
get
our
a quick
heading.
zoom
We're
on those 3 Gorgonians in the upper
need
left?
to go starboard
Stand by one, watch
toward
lead.
345. Right now I'm looking at 320.
Okay, the ship's not moving, correct?
No.
Okay, they watch lead wanted to check out these 3 over here. I'm gonna run over there. You can follow me, but before we start moving again,
Looks like more black corals to the left up here.
correct.
Yeah,
I guess I'll
requested a
confirm Nav.
quick
The
closer
ship
look.
is not moving.
Sure.
Yeah.
Keep following me,
It
Dan.
looks like 2 black corals and a
Beautiful spot.
Chrysogorgia. Yeah, there's lots of stuff here.
Ship is just about wrapping up its move.
Okay.
Okay.
And Mary, is that the same crab we saw on the other Antipitherian?
Okay, Dan,
Bridge
take
go.
control again.
It looks similar.
So move complete. Got
Let's
it.
do
What do you
this
need,
It
pilots?
sure looks like it.
First, go ahead
Awesome.
and zoom video
We will get you a zoom here shortly.
in the foreground, and then we'll do
Yeah.
background.
So we're
Much
going
as
to
we
zoom on
can.
the Chrysogorgia in the front.
Keep
There's
going.
also
Camera
a
down.
small yellow Gorgonian.
Ship move complete.
Copy that. Thanks.
Thanks,
Dan.
Nav.
Yellow.
There's Eurotychus again.
Eurotychus is the crab.
That's him.
Do we want to look at pink or yellow watch leads?
We've identified the pink one. I think the
Okay.
yellow one real quick would be better.
Sure.
That could be something overgrowing in Acanthogorgia,
come
the
down
bright
there.
yellow.
If you can.
Yeah,
Okay.
like zoanthid or something on it.
Like a zoanthid. Yeah.
Yeah.
And go for it, Ed. Ah, sorry. You can just hold there. I'll try to.
Yep. Yeah, the folks, experts in the chat room agree. That's a yellow zoanthid overgrowing part of a still living Acanthogorgia.
And the polyps may be swollen with
Come up a
eggs,
little bit, Dan.
according to Scott
Okay.
France.
Perfect.
If you quick zoom out, I saw something in the lower
Okay.
right
Come out.
when you're done.
Video.
The shrimp is Spacey Pellinella.
Go watch lead. Watch lead. What are we looking for?
The crinoid, I'm sorry, lower right, pink. Looks like it has a bunch of arms. Might be a starfish.
Okay. Yeah, hold there, Ed.
Yeah.
arcade video. See what we can do here. Start coming in. Okay, yeah, that sounds good.
This looks
Tilt
like the
for
same
me, Dan.
starfish we saw yesterday.
No, Dan, you got tilt down?
You got it.
There we go.
Beautiful.
I don't know if you have any more. Okay.
Did you guys want to take a quick look at the Antipatharians in the background?
Keep coming down a little bit, Dan. There we go.
This is the starfish.
Hey,
that
Stephanie,
what
I
we
see a
were
message
after
just
last
came in about the polyps might be swollen with eggs up above
Couple
that.
wide
On the
video.
Chrysogorgia, we can take
Above that
a look.
crinoid?
Let me direct them.
Might be worth taking a look, yeah, just in case.
Guys, can you? I have another request to zoom in on the pink gorgonium we were just on. If you could zoom in tight on that.
Okay. Sure thing. Let's get set up.